Well, today I'm going to give you an overview of proteins and amino acid metabolism. We have already gone through in detail regarding this chapter, but as far as examination point of view is concerned, I would like to give you certain guidelines how to prepare this chapter for your University of Health examination. If you look on that syllabus, which I have already given you the photocopies, and that is with you. And I told you, whenever you are going to prepare a topic, keep in mind, keep before you that university syllabus, because every question, everything is being settled there in the paper according to the syllabus. And second is the table of specification. I have already given these two things to you, and these are with you. As far as the weightage of this topic is concerned, it is a big topic and it has got a big weightage also. There are SCQ for 7.5 marks and MCQ 6 marks in this chapter. Will be asked for the university examination, and total it goes to 13.5 marks. So a very big weightage is there. So while preparing, you keep in mind that this is a very important topic, and you must go through very rightly through until the end of this topic. Now, how to prepare this topic? That is my second. I would like to divide the syllabus of this topic into three portions. And the very first portion, which is now you are watching on the screen, I have divided into three, and the number one is the overview of the protein turnover in human body, nitrogen balance, which will be positive and negative. And second is the intra-organs amino acid exchange in normal post-absorptive state. And this is how you will proceed. That keep this first portion, keep this first part in your mind. Now, the second, I have divided the second portion, the syllabus, into second portion, and that is the degradation of the amino acids. It is before you. While preparing, you start with the first one. Okay, then coming to the second portion. If you look very carefully to this portion, what important here is, it is all related with the ammonia in the body, its different toxicities and like that. So first of all, you see there is sources of ammonia in the body. Before starting this topic, you must go through the sources of ammonia in the body. Then second point is to keep remembering is the transport of the ammonia in the body. First is sources. What are the sources of ammonia? It's very simple. Then is the transport of ammonia in the body. And third one is the ammonia toxicity. This is also important. Different problems relating to toxicity of the ammonia. And fourth is the fate of ammonia in the body. Well, after going through all these four subunits, that the sources of ammonia in the body, transport of ammonia in the body, ammonia toxicity, fate of ammonia in the body, and very important then is the reactions and regulation of the urea cycle along with the metabolic disorder of the urea cycle. Then you go for, first of all, you go through ammonia, and when you will be familiar with that all, it will be very easy for you to understand another important topic, that is the urea cycle. And then there are certain disorders of the urea cycle. Now this is the second. I have divided your syllabus into three portions. This is the second portion. Now going for the next one, is third portion. I have made it third portion. Mind it, I have divided this syllabus part into three parts, three portions. Number one, I've shown you that is the regarding nitrogen. The second is the regarding amino acids removal and ammonia particularly in that. And now the third one is individual amino acids. You can watch here the glycogenic and ketogenic amino acids. You know there are certain ketogenic amino acids and the glycogenic amino acids. And when we go through to study all these, we got very important amino acids from examination point of view. For your attention, I would like to say phenylalanine, tyrosine, histidine, and then there are salient features of important metabolic defect in amino acid metabolism. Sometimes the examiner is very fond of giving these, that is phenylketonuria, maple syrup urine disease, histonemia, L-captonuria, homocystinuria, then tyrosinase and albinism. These are few important, though all are important, but I'm telling you, these are some of the important amino acids, their diseases mentioned here. We have to go through. If you pick up the university papers, previous university papers, you would definitely, you will find that there are questions, SCQ questions in this topic regarding phenylketonuria, maple syrup disease, then albinism, L-captonuria, the diseases and their causes, they usually asked in the examination. So, and similarly, there is the epinephrine and norepinephrine and melanin. These are important as far as the examination point of view are concerned. Now, to summarize, to sum up, I have divided this topic, the metabolism of proteins and amino acids, into three segments. You also divide when preparing it into three segments. It will be helpful for you. It will be no hodgepodge for you. You can easily then go through. The first is I've shown you the nitrogen concept. Then is the ammonia. Second one is name it with the ammonia, because ammonia sources, toxicity, everything is there, and ultimately the urea cycle. And then disorder, very important. Urea cycle is also very important from examination point of view. And then disorders of the urea cycle. And then for the individual amino acids breakdown. So this is how you will be going through while preparing this topic from examination point of view. Now, in the beginning, when I started with you the overview of the carbohydrate metabolism, I mentioned you that always keep in your mind the metabolic map. I'm stressing a lot on this point, and this is given in the book of Lippincott, page 92. I remind it again and again to you that it is given there in the 92. Now, this metabolic map very clearly is indicating that how, because the different amino acids, they are being taken up by the main cycle, that is the TCA cycle, from where there is integration of all these very clearly mentioned. And if you look, all this green portion here is indicating, written in the green color, that is all indicating your protein and amino acid metabolism here. If you see at the top, there is alanine, cysteine, glycine, serine, and threonine. They are then breakdown taking place. They are then converted into the pyruvate and you now know very well that pyruvate is then ultimately, when talking carbohydrate metabolism, the fate of pyruvate, and one of the fate of the pyruvate is that is when it is decarboxylated, it is converted into acetyl-CoA. And acetyl-CoA here the acetyl-CoA, now I would like to point it out, the acetyl-CoA is now said, and it will further then enter into the, it will, now this is marked red here, it is acetyl-CoA, and then it will go into TCA cycle, the tricarboxylic acid cycle, and you know here the complete breakdown, complete oxidation takes place here. This is one aspect I am showing you here from the metabolic map, that how these amino acids, they participate, individual amino acids, they participate in the metabolic pathway, and this is how the integration of the metabolic pathway is taking place. Similarly, if you look here, the isoleucine that is also being converted to acetyl-CoA, then leucine, phenylalanine, tyrosine, tryptophan, and lysine here, that they are all now showing you with this pointer that is all being then going to the acetyl-CoA, and that acetyl-CoA is then taking part. You know that, quite familiar, that is the point where it, with combination with oxaloacetate, it is entering into the TCA cycle, and TCA cycle where complete breakdown taking place. Like oxidation is also taking place. Similarly, here on the right hand side, there are certain other amino acid that is all, now you can see very clearly green color, and they are glutamate, but this glutamate and is then converted into alpha ketoglutarate, one of the part of the TCA cycle, one of the component of the TCA cycle, and this is how they then go into the cycle, TCA cycle here. After mentioning these individual amino acids, I would now like to draw your attention on the left hand side, where ammonia and carbon dioxide, I told you in the beginning that ammonia is important, sources of ammonia, its utilization, its then toxicity. Now here is the ammonia, it is in combination with carbon dioxide, it is formed into the carbamyl P, and this then entering into a cycle that is known as a very important cycle, that is known as the urea cycle. And this is, and I'm pointing it out, it is the urea cycle, and urea cycle itself is then other functions, but it can also enter, and it also enter into the TCA cycle via fumarate here. I'm all talking about this is to tell you how the integration of these two metabolic pathway, that is the protein metabolism and the carbohydrate metabolism, where they are being coming on a single point, and that is the TCA cycle. So definitely when we call of the metabolism, we call catabolism, and we talk of the anabolism. These are the two forms we have to see, and all these reactions very clearly indicating the catabolic as well as the anabolic pathways. This is how these amino acids, these proteins, they enter, and what is the role of this here of the urea cycle is going through that situation. Well, you see, definitely we know that the proteins, they are needed for the building material, they are needed for different reactions, they are needed in the body. But sometimes when there is a deficiency of these things, then these metabolic pathways, they are so integrated with each other that they help to coop up that efficiency. You have seen in the case of carbohydrate metabolism, I talked to you in detail regarding the gluconeogenesis. the formation of the glucose from the sources other than carbohydrate. And what are those sources? Definitely these amino acids, the protein, and talking of the fat also, that is the triglyceride. They are the other sources from where there is formation of the glucose that comes under the heading of gluconeogenesis. So whenever you study metabolism of any carbohydrate or protein or lipid, always keep this portion of the metabolic map, because this is very important. This can give you very clear picture how these different metabolic pathways, they are integrated, how they are linked with each other, from where they enter, and how they then move further. And this is what is we need to study when we are studying this all. Now, in your syllabus, I told you to divide this into 3 parts, and the first part is of your nitrogen balance. I talked with you, and it is mentioned in your syllabus also. When we talk of the nitrogen balance, we definitely be talking of the amino acids and proteins, and the best source of getting all this protein or amino acid is the animal source. That is the best one. And because why it is best? Because it contains all the essential amino acids. We need here amino acids while talking of the nitrogen balance in proportionate amount, and which is needed, required for the synthesis of human tissue proteins. If we see, I would like to draw your attention that while talking of the amino acids, classification of the amino acids, because we are now basically concerned here with the amino acids, there are 2 types. One is essential amino acids, and second is the non-essential amino acids. Obviously, you know that what are essential amino acids, and essential amino acids, there are certain amino acids which are not synthesized inside the body. You have to take in the form of the diet, and I told you the best source of the diet to take these amino acids is the animal source. So the plant source is also coming up to some extent, but the best one is the animal source. And when talking of the essential amino acids, I would like, you know that, I know, but I would like to remind you histidine, methionine, methionine, valine, isoleucine, and phenylalanine, and tyrosine, and lysine. These are all coming under the heading of the essential amino acids. This is given, this detail is given in the book of Lippincott at the page 262. I reminded, you can write it down, that the essential and the non-essential amino acids, that is, it is given very nicely in a chart at the page 262 of the Lippincott. And it has also divided these groups, these amino acids, into glycogenic and ketogenic amino acids. There are certain amino acids which are totally glycogenic. There are certain amino acids which are totally ketogenic, and then amino acids that are glycogenic and both ketogenic. You can watch this from the book on the page I have mentioned to you. Now coming to the nitrogen balance. You see, when in your first year when I was talking with you the nutrition topic, and I taught you that topic, there I mentioned you this very clearly, and for your recalling that, I am again showing you here that what is the nitrogen balance. Nitrogen balance, when amount of nitrogen consumed is equal to of the nitrogen excreted in the urine. And how it is excreted? In the form of the urinary urea nitrogen, or you can call it UUN. It is also excreted, that is one way. The second is sweat, and the third one is feces. and the most healthy adults are usually normal in nitrogen balance. You remember, I would like to recall you, I taught you when I was talking with you the role of the protein in the diet. There I taught you this, this just to remember you, because your present topic, that is metabolism, you need that knowing the background from where this nitrogen is being provided and what are the sources of the nitrogen. Then talking of we will talk that it is in balance. Definitely positive nitrogen balance and negative nitrogen balance. What is positive nitrogen balance? When nitrogen intake, I repeat, when nitrogen intake exceeds nitrogen excretion, that is known as the positive. Intake is more, excretion is less, it will be known as the positive nitrogen balance. In what situations? When, number one, growth, when the tissue growth occurs, you need nitrogen balance. For example, in childhood, there is tissue growth is there going on, so positive nitrogen balance is needed there, more protein intake and excretion less. Similarly, in pregnancy, you know that, and during recovery from the illnesses, when there is a recovery period from illness, definitely there should be positive nitrogen balance. Now, the second is the negative nitrogen balance. Now it is reversed to the positive nitrogen balance. In positive nitrogen balance, the nitrogen intake exceeds nitrogen excretion, whereas in negative nitrogen balance, nitrogen loss is greater than nitrogen intake. It is opposite to positive nitrogen balance. And when it is, this situation is when there is inadequate dietary protein. You must be now remembering I talked to you regarding the Kwashiorkor and marasmus there, and there was negative nitrogen balance because there was inadequate dietary protein. Lack of essential amino acids. I told you essential and non-essential amino acids. So lack of essential amino acids. Or during there is the physiological stresses. There are certain stresses such as trauma, birth, illness or surgery. When these conditions are there, there is likelihood of the nitrogen negative balance. A healthy person usually have a balanced situation. There is mostly that is commonly nitrogen balance. That is nitrogen, neither there is a positive nitrogen balance, neither negative. It is an equilibrium situation. How to calculate? How to find out? Nitrogen balance is given here. It is in your this. You can see I've mentioned here. The nitrogen balance can be determined by, there is a formula to find out nitrogen balance, that is the protein intake minus urinary, that is UUN, urinary nitrogen, urea nitrogen plus 4 grams. This is the formula. Why 4 grams? Because 4 grams account for the urinary losses in forms other than urea loss from skin and feces. So plus 4 is mentioned for the losses that is from the skin and the feces. And this is how we can find out the nitrogen balance. So this is also given in the page of in the Lippincott. What I am showing you here, that is in the book of Lippincott, page number is 367. Note it is on page number 367. It will help you to find out the nitrogen, because this is given in your syllabus. Now, another aspect I'm going to talk overall nitrogen metabolism. I've talked to you in detail. for that, and you can now, it is what I'm showing in this diagram is definitely all that nitrogen enters the body in a variety of compounds, and then amino acids important in the form of dietary protein. Nitrogen leaves the body as urea, ammonia, and other products derived from amino acid metabolism. Now this is the summary what I already now talked just now with you. The role of body protein in these reaction or transformation, two important concepts: amino acid pool and amino acid turnover. Sometime some may be questioned what is amino acid pool and what is amino acid turnover. Now, talking of the amino acid pool, this is definitely the pool word itself indicated. And for this discussion, the pool is supplied by three sources. This pool of amino acids or the pool of nitrogen, you can say, is supplied by three sources. Amino acid pool. Number one is amino acid provided by the degradation of endogenous body proteins. The word is endogenous. And second, three-way for the formation of the amino acid pool. Amino acid derived from exogenous. First it was endogenous, from inside the body. And there are certain amino acids, essential amino acids, and then non-essential amino acids coming in action here. The second is the amino acid derived from the exogenous, that is the dietary protein. First one is the protein from the body. The second is now from the dietary protein. And third one, that is non-essential amino acid synthesized by the simple intermediates of the metabolism. Very rightly said that this pool, amino acid pool, is formed by three sources: amino acid, which is derived from the endogenous, that is the body protein; second, which are exogenous, that is the dietary protein; and third one is the non-essential amino acid, which are going through the intermediates of metabolism. And now this pool is formed by three ways. How now this pool is depleted? It is also now depleted by again three ways. Formation of this pool is by three ways, and depletion again is by three routes. What are those routes? Number one, synthesis of body protein. Very true. Synthesis of body protein. Number two, consumption of the amino acid as precursors of the essential nitrogen containing small molecules. And third one is the conversion of amino acid to glucose, glycogen, fatty acid, ketone bodies. I told you when I was showing you that map, there is gluconeogenesis, there is how these are broken down. I mentioned you there. And this third one, that is the conversion of amino acid, that amino acid can also be converted into glucose when there is not prolonged fasting for many days. Because your brain and your other body organs, they need sugar, they need glucose. And from where that will be provided? Of course, firstly, it is provided by the store of glucose in the body in the form of glycogen. But that source is finished, then that force is lifted for a limited period, then definitely that glucose will be supplied by the amino acids. Then fatty acids, then ketone bodies, they all provide this. So this is how there is that pool will be then utilized. So three ways to formation of the amino acid pool, and then three ways to this depleted by 3 routes. Now, in the healthy, well-fed individual, the input to the amino acid pool is balanced by output. Similarly, as in the case of the negative nitrogen balance and positive nitrogen balance, here is in the healthy, well-fed individual, the input to the amino acid pool is balanced by the output. That is, the amount of amino acid contained in the pool is constant. Amino acid pool is said to be in steady state, and the individual is said to be in the nitrogen balance. This is what I want to point you out, how the nitrogen balance is then taking place.